This is our presentation on the photosynthesis of sun and shade-adapted plants by Cole Shortall,
Maya Greaves,
and Julio Fernandez.
So to start off, I'm going to talk about light intensity in relationship to photosynthesis. So light intensity is a rate-limiting factor in the process of photosynthesis. The amount of light provides a number of potential complications for a plant species when it's growing. If there is too much light, a plant could lose too much water due to evaporation. It is also possible that the plant produces an overreactive oxygen gas compound which can harm the plant. On the shade side of things, if a plant receives too little light, it will not be able to produce enough energy to survive. And as a result of this, plants must adapt to their environments, whether it be sun-abundant or shade-abundant environment. Each plant species also has different morphological features that are adapted to its specific environment. So condition-adapted plants evolve through generations of living in shaded environments, and plants with successful traits are able to produce offspring and spread those favorable traits. And in addition to this, plants often adapt their photosynthesis to their light conditions. So shade-tolerant plants have adapted their photosynthesis to function optimally under low light conditions, and these plants therefore are capable of long-term survival in shaded areas, and in contrast, plants that prefer sunlight adapt their growth to receive maximum sunlight. In this experiment, we studied different photosynthesis between a shade-adapted and a sun-adapted plant from Tecolote Canyon. We measured the stomata count, the chlorophyll concentration, protein concentration, and photosynthetic rate of one shade-adapted plant, one sun-adapted plant, and one plant whose condition is unknown. So using this data that we collected, we can infer to which condition our unknown plant is adapted. So we chose a sun and a shade plant from Tecolote Canyon. The south-facing side receives more sunlight, so sun-adapted plants thrive on this side, and the north-facing side receives less sunlight, so shade-adapted species are more abundant on this side of the canyon. And so lemonade berry is most abundant on the north-facing slope, so it is a shade-adapted species, and black sage is most abundant on the south-facing slope of the canyon, and so it is sun-adapted.
Our unknown plant is the cabbage tree. This tree is a palm-like subtropical tree with sword-like dark red leaves. It thrives in deep, fertile, and well-drained soils. It's native to Australia, so it is also viable in a drought climate and in parts that border the cooler parts of the country. We hypothesized that the cabbage tree is going to be a sun-adapted plant species because it's native to southern Australia, which means it most likely receives abundant sunlight. We also hypothesized that the measurements for the photosynthetic rate and rubisco concentration will be lower, as the chlorophyll concentration and stomata count will be higher than those of the shade-adapted plant species. Our driving research questions are: how do the native plant species, black sage and lemonade berry, differ in their photosynthetic capacity, as well as, is cabbage tree a sun or shade-adapted plant species, based on the information from the data of the lemonade berry and black sage plants we measured before? This research question is interesting and important. It is interesting because the species are specific to their side despite many differences. They're specific to their own side, even though their own sides are only about 100 meters apart from each other. And yet they still note incredible morphological differences. And it's important because each species can only survive on its respective side. This means that if you took a sun-adapted plant and put it on the opposite side of the canyon, it would not be as viable for survival because it is not adapted to that environment.
So for methods and material, we first started out with measuring chlorophyll concentration, and to use that we used a chlorophyll meter, in which we attached a meter to 3 different levels on each plant and made 3 measurements per plant for a total of 9 measurements. When measuring leaf count, a group member carefully counted the number of leaves on the plants twice to ensure the second number was counted accurate, because the plants are very complex with their leaves and we want to avoid human error as best as we can. So for photosynthetic rate, to quantify it, we used an infrared gas analyzer, and we made sure our C values were correct before recording the photosynthetic rate, and we chose a leaf in the upper middle section of the plant that can fill the whole chamber and waited 30 to 45 seconds until we recorded our data, and then we repeated that cycle. When measuring the rubisco concentration, we first conducted a Bradford assay using bovine serum albumin, a protein starch solution, to generate a standard curve. From there, We took each of the 3 plant species and punched 6 holes in them, making a total of 18 discs. So after grinding the leaves, we vortexed them for 3 seconds and we incubated them for 30 minutes in room temperature, then centrifuged all the tubes for 10 minutes. During this time, we prepared 1 test tube for each microcentrifuge tube, and then after that time had passed, we used the same spectrophotometer protocol from creating the standard curve to take the absorbance of 6 lemonade berry, black sage, and cabbage tree solutions. So the purpose of taking these parameters, more specifically chlorophyll concentration, was to measure how much light was absorbed and transferred to energy storing molecules that undergo photosynthesis. With stomata count, the more carbon dioxide that can be absorbed and water that can be released amplifies the potential for control of water loss and carbon dioxide uptake. Hence, high amounts of stomata indicate fast growth and wet climate, whereas lower amounts of stomata indicate lower rates of photosynthesis. The photosynthetic rate was measuring each species because the values showed which plant is undergoing the most photosynthesis and at what light intensity. We can use these values to compare how shade and sun-adapted plants differ in their photosynthetic capacity. Robusco concentration is directly correlated with the rate of photosynthesis as well.
Next up is our results section. So first we have chlorophyll concentration, which is shown in this graph, is the average concentrations for our 3 plant species, our 3 measurements. For black sage, the sun-adapted plant displays the highest concentration of chlorophyll with 557 milligrams per meter squared. Cabbage tree displays the second highest concentration with 500 milligrams per meter squared, followed closely by lemonade berry, which is 446 milligrams per meter squared. And the error bars on this graph show the standard error of the mean, and we ran a one-way ANOVA. This test did not show any significance between any of the plant species, as all of the p-values were greater than 0.05. Next is stomata count. For cabbage tree, it ranged 4 to 6 and averaged 5. For lemonade berry, it ranged to 93 to 105, averaged 100. And for black sage, it ranged 17 to 25 and averaged 20. And in this graph here, we see all these counts together. Lemonade berry had the greatest average count at 100, black sage second with 20, cabbage tree third with 5. And the error bars represent the standard error of the mean. And the one-way ANOVA test we ran showed an extremely significant difference between all 3 species, with all of their p-values being less than 0.01. And to show this extreme significance on the graph, we used 3 of each significance sign. So we used 3 money signs to show significance to lemonade berry and 3 star signs to show significance to black sage. Next is photosynthetic rate. This top graph shows photosynthetic rate compared to light intensity for the 3 plant species under 8 different light intensities varying from full shade on the left to full sun on the right. Lemonade berry had a higher photosynthetic rate than black sage and cabbage tree, whose values progress very similarly. The maximum photosynthetic rate is 12.66 for lemonade berry, 8.3 for black sage, and 7.7 for cabbage tree. And the graph underneath this is showing quantum efficiency. It's the first 3 points of our photosynthetic rate graph. And the curve for lemonade berry in this stage has a continuous upward linear regression. Black sage had a great upward increase compared to the gradual progression of lemonade berry and cabbage tree. And the quantum efficiencies for these 3 plant species are 1.8 for black sage, 1.5 for lemonade berry, and 0.8 for cabbage tree. And the results for our last parameter, Rubisco concentration, this graph shows the amount of Rubisco in milligrams per milligram of leaf tissue. So lemonade berry had 0.043 milligrams, black sage had 0.066 milligrams, and cabbage tree had 0.03. Black sage, the sun-adapted plant, had 2 times the amount of Rubisco as cabbage tree, and definitely less than lemonade berry as well. And we ran a one-way ANOVA, and this test showed extreme significance between the concentrations of all 3 species, with all p-values being less than 0.01. And to show this on the graph, we used hashtags to represent significance to black sage and stars to represent significance to lemonade berry, and we used 3 of each sign to show that the significance was extreme.
For our discussion, we went back to our original hypothesis, which was how do the native plant species, the black sage and lemonade berry, differ in their photosynthetic capacity. In our results for photosynthetic rate displayed previously, we concluded that the lemonade berry, the shade-adapted plant, had a higher photosynthetic rate. The black sage and the cabbage tree, the both sun-adapted plants, had similarly low photosynthetic rates. A possible explanation for this finding is that the shade would require a stronger ability to photosynthesize under its limited light conditions. At any concentration in micromoles over meters squared per second, the lemonade berry is more effective at photosynthesis. This makes sense because it lives in lower light conditions, meaning that in order to compete for equal energy with the sun-adapted plants, it would have to have a higher rate of photosynthesis. Is a cabbage tree sun or shade adapted? We supported our original hypothesis that the cabbage tree was a sun-adapted plant. The data collected in this experiment supports this hypothesis because all of the parameters, excluding the rubisco concentration, show significant similarities between cabbage tree and black sage, the plant we know to be sun-adapted. The similarities in photosynthetic parameters between the black sage and the cabbage tree support the hypothesis that the cabbage tree is a sun-adapted plant. Both stomata counts are relatively low compared to the lemonade berry, and this is a known trait of shade-adapted species. Zooming out to the bigger picture, when we examine other plants, we can identify each of them as sun or shade adapted, as other students did with this experiment, meaning that it's not just limited to our application with the cabbage tree. For the understanding of implications of sun adaptations and shade adaptations, we realize that each plant will thrive in its own specific environment with its own conditions. And as for the real-world applications, if a farmer, for example, wanted to maximize crop output or yield, they would benefit from the knowledge of whether their plant is shade or sun adapted. If their plant were to be shade adapted, they could place it in lower light conditions so that it can maximize its photosynthetic rate and energy budget. And these are references which we used throughout the experiment.